transport. And so we'll go through the passive transport, active transport, concentration gradient, and then this large molecules in and out of the cell. So we know that molecules move down their concentration gradient. That's passive transport. That is, it does not need energy to do that. So net movement of molecules from high concentration to low does not need metabolic energy, so ATP is not required. And it's important for passive transport to happen so that molecules can move from high concentration to low. When the cell has too much waste product, it wants to get rid of it. When there is too much water, it wants to get rid of it. When there isn't enough water, it wants to take it in. Right? So passive transport plays a large role in the survival of the cell. So there's constantly molecules going back and forth between the inside environment and the outside environment of the cell. The types of diffusion, we already know a little bit about it. So we have the regular old passive transport, that's regular old diffusion, without any help. Small nonpolar molecules have no problem getting through. Nitrogen, oxygen, carbon dioxide gases, they're constantly moving back and forth without a problem, which we want, right? We want it for the survival of this cell. Then, of course, we have facilitated diffusion. These are still going high to low. However, the molecules either have a charge, so they're either polar, they're charged, or they're too large. So if it's non-polar, but it's large, then it's still not going to be able to get through without some kind of help. Okay? So, facilitated diffusion, both the carrier proteins and the channel proteins' job is to get molecules through that normally cannot pass through without help. And again, that includes polar and charged molecules, and it includes large molecules. The polar and hydrophilic molecules, right, can't get through the cell even if they're small because the tails don't like it. Ionic compounds, anything with a charge, can't get through unless it goes through a channel protein or a carrier one. Or in some cases, it's active transport we'll look at later. Okay? Now, when we talk about active transport, what is the difference between active and passive? What's active doing that passive is? Yes. Against concentration gradient. That means it's going from low uphill to high. That is a difficult thing to do because that's like thinking about going uphill process, right? Anything that's uphill is going to need an input of energy. So, active transport uses ATP as its energy source, and it moves ions or other molecules against their concentration gradient. And there are specific ones that do this that are very important. There's a sodium-potassium pump that is very important for nerve function. Without that pump, your nerves wouldn't function properly. So it is a very important process. Then, of course, we talked about those large molecules. Remember endocytosis and exocytosis, right? Pino and Phago. In endocytosis, the cell uses energy to take macromolecules. So now you're looking at a large molecule. And a large molecule requires a little bit more. It does require energy. It's not enough for it to go through a channel. So macromolecules and particulate matter, it forms new vesicles from the plasma membrane, and formation of a vesicle requires energy. So in endocytosis, we have two different types, well actually three different types. The third one you haven't seen until now. You have phagocytosis, which is large particles, chenocytosis, which is extracellular fluid, okay, so liquid, and then we have receptor-mediated endocytosis, where the receptor protein on the cell membrane is used to capture the target molecule. So what has to happen here, if you pay attention here, there are receptor molecules. Receptor molecules are molecules that are on the surface of the membrane that's going to grab some kind of molecule. Okay? When it grabs it, then it's going to allow for something to happen. Usually it's a vesicle formation. But once it binds to it, it allows for the vesicle to form, and then we call that receptor-mediated endocytosis. It's still taking large moleculs in, but there's an extra step. There's an added step before it makes the vesicle. It's a receptor binding to a molecule. A molecule binding to a receptor. Then it does what the normal endocytosis would do. Okay? Sure. Receptor molecules. Receptors are things on the membrane that are going to bind molecules. Receptor-mediated endocytosis is when something is binding to a receptor and it's mediating. That means it's allowing for the vesicle formation to happen, for endocytosis to take place. Okay? It's still endocytosis, but it's called receptor-mediated because you have little receptors, sites, and things bind to these receptors, and then that allows for the vesicle to be formed. There's like an added step before endocytosis can happen. Okay? Yeah. Certain molecules go through that because then they're going to go and bind somewhere else. So it gives it a little bit more specificity to what it's doing. Okay. Then we have exocytosis, same concept. Now it's taking it from inside the cell and out. This includes proteins such as signaling proteins, hormones, waste product. So hormones are very important because, again, remember I talked about this yesterday, you make them in such small amounts, yet it has a huge effect on the entire body. And so where they're made is not enough just for them to stay there. They have to be sent off to other places, and we'll look at that later when we talk about signal transduction pathways. Okay? All right. Let's see what kind of experiment they're doing here. A scientist discovers a freshwater species. It's called Acanthaphia polyphaga in a local pond. This little single-celled organism, known to use endocytosis to eat smaller single-celled organisms like bacteria. The scientist takes a water sample from the pond to study this little organism. Several bacteria are added to the water sample. The initial number of bacteria added and the final number remaining after 30 minutes are counted. What could the scientist propose as a next investigation to determine whether this little organism used endocytosis to engulf the bacteria? It's trying to figure out if this thing is doing endocytosis, and it's doing endocytosis apparently to take in smaller bacteria to engulf, and they're counting out the number of bacteria before and after, like in 30-minute period. And the question that's being asked is, what other things can they do in order to determine that this endocytosis of the bacteria is actually be a method. So it gives you those 4 options. Count number of ribosomes before and after. Count number of vesicles before and after. Count number of bacteria that swim away. Count number of the organism present before and after. Which one do you think? Vesicles? B? Okay. Does everyone agree with B? Let's see if you're right. No, you want A? Right? Why are we counting vesicles? We just said it's endocytosis, right? So that means it's going to do what? It's going to engulf using a vesicle, so that's the one way you can determine. Okay, so passive active transport, selective permeable, exocytosis, endocytosis. We're going to stop there for today. And we are going to continue, and then we'll start osmosis on Monday.